So this first chapter, a lot of it has to do with general biology, and so like I said, it should be reviewed. I'm going to go really fast over a lot of it. Some of it, you have to realize that this is from a secular perspective, and so it's there for you to know. It's not necessarily saying that it's not. None of it's necessarily any ideas that's endorsed by the university or any particular individual at the university. Okay, and so obviously, as I was just saying in Intro to Chem, all material, actually, it's not even just living things, are made up of molecules. In particular, we talked about certain specific biomolecules here. We all require the use of energy. We have the whole idea of the law of conservation of mass and energy, which is where matter or mass and energy are neither consumed nor created nor destroyed in a reaction. Okay, one of the reasons why I like biochemistry is now we can start to see some of the reasons why you needed to learn Gen Chem and hopefully organic chemistry and put those into a living context. Okay, plus it's just really cool. And but yeah, there are certain small molecules, and so the idea, you've had life origins, or most of you have had life origins. That the simplest molecules are those that can be comprised to make larger molecules as you go along. Okay. Then we have this really high class organic chemistry, had to do with carbon. So everyone had to take organic chemistry. I'm sure you all loved it. Just thought it was the most amazing thing ever. What? What was that?
I liked it better than Gen Chem.
Oh yes. I mean, I know. Whenever I took Gen Chem, it was 3 semesters. Here's his 2 semesters, right? And so, but yeah, I actually took organic chemistry 1, and then my organic, even at the University of Missouri, huge university, they still have class scheduling conflicts. So organic chemistry 2 is done to conflict with biochemistry, like the first real biochemistry class, and so I put it off like a year, maybe a semester or 2 semesters maybe. So I took biochemistry, which I thought biochemistry was a little bit more difficult than what it should have been. Then I went back to organic chemistry 2 and thought it was a breeze. I didn't study it. Because what you're going to find out, hopefully, is a lot of, especially when we get into metabolic pathways, we're talking about carboxylic acids and amides and all this kind of stuff that you use for organic chem 2, and so that's why it's really pretty simple. But the same rules apply in vivo that apply in vitro with respect to organic chemistry. We see the same kind of biomolecules and the same type of functional groups. The big difference really has to do with the word I'm looking for, the conditions, right? Because to go from an alkene to an alkane in organic chemistry, you'd say, oh, it's hydrogen H2 with platinum, palladium, or nickel. Well, we don't have platinum, palladium, or nickel electrodes in our body, so we have to find some other way to do that. But it's the same type of principle. The functional groups behave the same way. A carboxylic acid is still much more stable than what you'd expect because of the fact that there's a resonance structure. So we have carboxylic acids. Carboxylic acids are hydrophilic. Okay, that's so therefore they behave a certain way. And so we see the same things over and over and over again. Okay. So this is just some examples of biomolecules that we can see. Let me get to a little pointer here. So we have nucleic acids, which are nitrogenous bases. We've got sugars, which are, let's do a review real quick. There's an R group, OR. So what kind of functional group is ROR? Ether, yes, awesome. And we have this group right here. So what kind of functional group is this right here? This OH? Alcohol. Okay. Then we also have the lipids. There's the carboxylate. Remember, just to do a quick review, because I'm not for sure. I would always reinforce this in organic chem 2. I don't know if Dr. Salvatore did or not, but there is a difference between carboxylate and carboxylic acids. So most in vivo, most of the will be in the form of carboxylate. Okay, this is where this carboxylic acid lost its hydrogen, so it's negatively charged. So the carboxylate end, and then this really long lipid chain. So there's OVH, palmitate. Okay. And so, but you can they're still going to have very similar properties versus what you would think of, in vivo versus in vitro. And so, like if we look at palmitate, What's something that you can, what's one physical property about this? Is it going to be hydrophilic or hydrophobic here? It's very hydrophobic. Okay, it's got one little. It's called being amphiteric or amphiphilic. It's got the carboxylic acid in the carboxylate can hydrogen onto water, but all this is very hydrophobic. Okay, there's a glycerol. Glycerol is one of the few that you're going to ultimately memorize its structure. Okay, not so much this semester. Next semester, it's almost like a Miss America kind of answer. Okay, because it's a common intermediate in many things, or its derivative. So if you can draw the glycerol, which is just 3 carbons with 3 alcohols, okay, it's very simple. That's the backbone of triglycerides, monoglycerides and diglycerides. Glycerol, you turn one of them into an aldehyde, you make glyceraldehyde. You turn one of them into a carboxylic acid, you make glycerate, okay, which is important. You find out that you just turn certain things on the glycerol backbone, and that's why you'll see glycerol over and over and over and over and over again. You'll probably get tired of it almost, but glycerol is a very common one. And then we have codeine and so on and so forth. But this just shows you these. These are organic, with the exception of phosphate, these are all organic compounds that are important in vivo. Phosphate is a functional, you guys didn't cover phosphates in organic chem too, right? Usually we didn't ever have time, so I doubt that Dr. Salvatore had time. Did you cover sulfur at all? What? Thiophthalates. Did you cover very much? You probably didn't cover thioesters. Okay. And so, I know. How do I, there we go. All right. And so we're going to talk about those. These are important functional groups in vivo and biochemistry. Oh, then we have things like amino acids. I will tell you right now, you will need to memorize all 20 naturally occurring amino acids. You need to know their structures, their names, their 1-letter codes and 3-letter codes. Write it down. It will be on a quiz. And then once we cover that for that quiz, there's something about the amino acids on every single quiz and exam from here on out for Biochem 1 and Biochem 2, provided that I'm teaching it. Because it's essentially the alphabet for biochemistry. Okay? And so we're going to go, chapter 3 is amino acids. We'll go into great detail. But you may want to start memorizing it now. In the book, there is a table, not this one. It's really good. I forgot. I used to write it down. It's in chapter 3, I think. But it has them all listed out. Okay? And it has them separated out according to their functional properties as well. But start memorizing now. There's 20. You only need to know the 20. I should say there are 20 naturally occurring ones that are made to use protein. You'll find out later on there are even more amino acids. But the reason why they're called amino acids, why are they called amino acids? If you look at these examples, what? There's an amino group. Whoop. And what's the acids? Carboxylate. Okay? What kind of, this is something I cover in organic chemistry too. Once again, I don't know Dr. Salvatore's background, if he's biomedical or not. What kind of amino acids are these? Does anyone know? Because there are multiple kinds. No, they are, but they're all going to be polar. These are called alpha amino acids. Does anyone know why? Because I don't know if you went over the nomenclature, like the alpha, beta, gamma, delta, epsilon, sigma. Pardon? It's on the alpha carbon. Okay, so the way that I would always explain it, so this is a carboxylic acid. Okay? So when you draw it out, oh yeah, that's fine. I'm going to draw out just the carboxylate. This is not the official term, but this carbon right here, the one that's this is called the carbonyl carbon. It's also called an acyl carbon, and it's also called the carboxyl carbon. But this one, just in case I call it one of those, this carbon I would always refer to as ground zero. Okay, so the carboxylic acid, since it's making 3 bonds to oxygens, it has a higher priority than anything else right there. So it's got the top priority. The carbon next door to it is called alpha. They started using Greek letters for numbering, alpha, beta, gamma, delta, so on and so forth. And so the carbon next to it is called C alpha. So that's why, if we look at it, doesn't matter. All of these are alpha amino acids. So this would be ground zero. That's the C alpha. Hanging off the C alpha is the amino group. Okay, there is a type that's also biologically important called a beta amino acid, and that means that there's going to be one more linking carbon in between, because the amino group would be hanging off the beta carbon. But those that are used to make up proteins are all alpha amino acids. Okay, so it's this little, like I said, since I didn't have you for organic chemistry 2, I don't know how much of that verbiage that is used. So biochemistry uses it all the time. They love their Greek letters. Okay, alpha, beta, gamma, delta. And so usually you don't have to know beyond that. Epsilon. First 4 or 5. All right. What was I doing here? That's always a good one. Oh, these are just functional groups, and typically, did it get cut off? Let me see. Nope. We just reviewed like some of these functional groups, and so on your own, these are all of them except for possibly these over here. You should know. Okay, so for example, this one right here. What functional group is it? It's a what? Thiol, okay. Then we already went over ethers. What functional group is this one right here? Alkene. What about this over here? Ester. R group, carbonyl, OR. All right, versus this one, the one right below it would be an amide. Okay, and in biochemistry, by the way, they also call it an amido group. AMIDO, amido. Same thing with an amine or amino, like amino acid. So just to get you, we'll talk about thiols in a little bit because thiols are a humdinger. Okay, but then we have these two that are down, and that's a scientific term. No, we have these two down here. What do you suppose this functional group is called? If you just had a look at it, it's a phosphoryl something, or phosphoric or phosphoryl. What do you suppose it would be? What is it kind of like up in here? Ester, and this is called a phosphoryl or phosphoric ester. Okay, so this is where the P takes the place of the carbonyl carbon. This right here, actually the carbon derivative of it's not on there, but what do you suppose this is? This is the phosphoanhydride. When there are R groups hanging off of it, it's called a phosphodiester, which is what the backbone of DNA is, right? The phosphodiester. Well, except for DNA, it only has one phosphate, but it's the same principle. Just like when you have esters and diesters, you can have that with a phosphodiester and phosphoester, phosphoanhydride, all that kind of stuff. Okay, so that's why I'm going to... This is actually from your book. I don't know, since this is from the earlier edition, the actual number of the figure may have changed. These, if you notice, you have to memorize these. There are certain things you have to memorize. You have to memorize your amino acids. You have to know your functional groups. A lot of these you already know because they're just continuation from organic chemistry. However, there are some that I doubt that Dr. Salvatore covered because typically most organic chemistry books will wait and put those maybe at the very last, like chapter 30, you know, that kind of thing, because when they start talking about biochemistry. Or they didn't put them in that one table that I always had you memorize, and he probably told you to have too, like early on, like chapter 4. What it is if you look, I'll just point at the ones that are different. Of course, we know methyl and ethyl. You know, make sure you still know your prefixes. Meth, eth, pop your butt. 1, 2, 3, 4. Meth, eth, prop, ut, pent, het, het, oct, non, dec. Those are still used in biochemistry. This is a phenyl group. It's really important you know the difference between phenyl and benzyl. Does anyone remember what's the difference between a phenyl and a benzyl group? That's something I would always stress in organic chemistry. In fact, there's a whole chapter on. Nope, they're both benzene rings. It's whether or not it's attached. If it's directly attached to something, it's called a phenyl. It seems completely backwards to me in organic chemistry. If it's directly attached to something, it's called a phenyl group. If there's a linking carbon and then it's attached to something, it's called a benzyl group. Okay. In biochemistry, we only usually talk about phenols. Okay, that's it. And we're going to find out that phenylalanine is a benzene ring that has a linking carbon, so technically it's a benzyl group. But it's called phenylalanine. And the reason why it makes sense once you learn that, because it's alanine, it's got a phenyl group attached to it. So once you memorize your amino acids, it'll make sense why it's called phenylalanine. All right, so then we have our aldehydes and ketones, which are the carbonyl groups. Carboxylate. If you notice, they don't even show carboxylic acid because the fact that the only difference is the presence or the absence of the hydrogen. Alcohols are also called hydroxyl groups, and they talk about what you call them hydroxy, if you remember right, whenever you named them in organic chemistry. Enols, you cover that in organic chemistry, ethers, esters. Acetyl groups are so important, and there's something in vivo. There's something I want to point out. This is acetyl, which is very similar to acyl. Okay, acetyl is a specific type of acylation. So acyl looks like this. R, O, carbonyl, R prime. Okay. Whereas acetyl, that R prime has to be a methyl group. Because I don't know if it's an old term, if Dr. Salvatore talks about that, but the term acet, A-C-E-T, means two. And so that's why you see acetic acid is a carboxylic acid, it only has two carbons. Acetaldehyde is an aldehyde, it only has two carbons. Well, acetylation, which is a functional group in biochemistry, means that there's only two carbons total. Acylation, which is very important next semester, means that it can have more than two. It could be two, but it could be a lot more than two. It's a generic term. Anhydride is the same. Amino is the same thing as an amine. Okay, it's just that in biochemistry we use the O part. Whoops, I keep hitting the wrong one. So that's amino or amine is the same thing. They're showing it here being protonated. Amide and amido are the same thing once again. This is one that is covered in organic chemistry. At least I would always cover a little bit in organic chem 2 and let you know, this is really important for later on. It's called an imine. Imine looks just like a ketone, except it's a nitrogen. And remember, nitrogens can make three bonds, so that's why there's the hydrogen on there. There can be another R group depending upon the type of, instead of a hydrogen, depending upon the type of imine. You're going to find out, right here it is, that these are very important. Some of you guys, when you pick your enzymes, they may even use what they call a Schiff base, which is just an imine that's got another R group instead of hydrogen. This one's probably brand new, right? I'm sure that you guys didn't talk about a guanidino group or guanidinium. If it's positively charged, it's called guanidinium. If it's neutral, it's called the guanidino group. I doubt that. Does anyone know why we have to know about it in biochemistry? What does it look like? In particular, it's for one amino acid. It's arginine. Okay, and then imidazole is new. Okay, imidazole looks like the imidazole group is what makes up histidine. This is where I always point this out because this right here is called a thiol, and we usually talk about, in biochemistry, I call it thiol, and a lot of people call it thiol too. They also call it sulfhydryl. They also use a term called mercapto. Like, especially in naming, it sounds really weird. Like, beta mercaptoethanol, BME, it's something you use in biochemistry lab or in biochemistry all over. And it's just a thiol. Okay. Whenever it loses its hydrogen, what's it called? Thiolate. And that's very important, okay? When it looks like this, it's negatively charged, and it's called a thiolate. It's used all the time in vivo in biochemistry. I don't know if you guys covered a lot about thiolates in organic chemistry, because usually in a lot of organic chemistry books, it's just like a little page, a little blurb. But what's so important about sulfurs? Sulfur's used a lot more in vivo than what you guys did in vitro for organic chemistry. Because organic chemistry uses a lot of oxygen, because it's in the air and it's a little bit. What's so special? You may have to use the periodic table. What's so special about sulfur as compared to oxygen? It's what?
Or you can expand the octet, or it can block.
It can. Mm-hmm. We don't use it all that time for that reason, but it can expand the octet. For sulfates, when you're doing sulfates and sulfites, that's true. What else is it better at?
A good nucleophile?
It's an awesome nucleophile. Okay, then oxygen. What else is it? If it's a good nucleophile, it's also a good
Leaving group?
leaving group. It's an awesome leaving group. Okay, it has to do with pKa's. And so we'll talk about that later on. But that's why your fats, for energy or for storage, you do the enzymes that are involved utilize sulfurs, which we're going to find out that it's amino acid cysteine. Okay, and the reason why is because it's better than oxygen going in and attacking and being reversible. That's important. You know, just because something's a good enzyme can do a good nucleophile, you've got to make sure that it's reversible, or otherwise you're going to die. Okay, so I talk about dying a lot in biochemistry. You can either think of it as being pessimistic or think of it as being a miracle of God, miracle of life, but you can die a lot. Okay, we have disulfides, we have thioesters. Thioesters are hugely important. You'll see thioesters a lot next semester. And it looks just like an ester, except it's got a sulfur. I used to point that usually in probably the ester chapter of RNA chemistry, they have like a little, almost like a little box sometimes that just talk about like biological importance. And they may mention thioesters, but they used to talk a lot. Then we have the phosphate stuff. Okay, phosphoryl just means it's a phosphate that's attached to something. Phosphoanhydride, we're talking about that. And a mixed anhydride is when you can have either carbon and a phosphorus on each side. Okay. All right. Yes. Oh, this is just where I used to blow it up and take even longer going over all of them. Okay, so this is where we get to practice. I picked these two molecules first. Give me a moment to kind of looking at things. Don't cheat if you look and know the answers. The first one is dihydroxyacetone phosphate, which has, it's another one, it's like glycerol, in fact, it's based off of glycerol. I don't know if you guys can see that. This is actually one of the glycerol derivatives. Because how many carbons does it have? 3. And so you're going to find out 3 is like a magic number. I think it's like the Trinity. No, I don't know. Faith integration sometimes. I push the limits. Okay, but it's called dihydroxyacetone. What does going from organic chemistry, what is acetone? Like, how do you draw acetone? How many carbons does it have? 3. And what makes acetone acetone? It's a ketone. The middle carbon is in double oxygen, okay? And once again, it's the simplest ketone, a ketone of 2 carbons, so it's where it's acetone. Dihydroxyacetone phosphate, which literally means 2 alcohols on the ends, so 2 alcohols, and then one of the alcohols has been converted to phosphate, so I kind of give away for what functional groups there are. This one's really important. It's a common metabolite in lots of different metabolic pathways, and they even give it a special name, which to me always sounds like it's like a hip hop kind of name, or DJ name. You know what they call this one? It's DHAP, D-H-A-P, DHAP. So, that kind of thing. All right, so this is DHAP, this is ATP. So what's one of the functional groups on ATP? Anyone, what's a functional group on ATP?
Ether.
We have an ether. We have R, O, R. What else do we have? We have an alcohol. Well, actually more than one, but we have alcohols. Okay. And what are these? What's this? It's phosphate, phosphoryl groups. What about this one right here? Yes, yeah, this is a type of imine. See, there's a carbon double bond to a nitrogen. Okay, then we have this one right here, which is an amine. And what about this one right here? This nitrogen. What kind of functional group is it? It's just an amine, because it's a nitrogen with 3 single bonds hanging off of it. Okay, but yeah. We want to do more. This one's already, the answer's already given to you. This is huge. This one, oops, oh, I did the wrong one. There. Does anyone, this is acetyl-CoA. So this is an acetyl group. It's actually not even listed, but there's an acetyl group right here. Okay, so that's acetyl-CoA. What, first of all, does anyone know what is acetyl-CoA used for? What's one of it? specifically what's acetyl-CoA used for. It's kind of in the name.
Is it used for the Krebs cycle?
In a way, yes. It is involved. Well, not acetyl-CoA. It's not involved directly. You're going to find out. That is one of the ones that I get to use that has that space integration. It's Moses in the Holy Land. So that's why there's a little asterisk. That's why I kind of hate to name that one specifically. Coenzyme A, which also has a really cool. Does anyone know how they abbreviate coenzyme A? They call it CoA, first of all, but what's the other abbreviation for it? It's also kind of hip-hoppy. It's called CoAsh. Okay. No, acyl coenzyme A, the acylated coenzyme A is involved in acetylation. So it just transfers all. That way it's on the video. Like what it does. This right here, we have all of this. You see, amido groups, hydroxyl groups, phosphoanhydride. It goes on and on and on and on and on. The business end for coenzyme A is that sulfur. The rest of it's just window dressing. Okay, so that sulfur, when it's coenzyme A, free coenzyme A or CoAsh, it's a thiol. That's what the SH stands for. When it's acetylated, it's because that sulfur went and attacked something that had an acetyl group on it, and then it's easily attacked right here, which I assume that you guys went over tetrahedral intermediates and stuff in organic chem 2. So what you do is you smack it, sound effects, I was just say sound effects optional, boom, it makes it tetrahedral intermediate, you get collapse of the tetrahedral intermediate, and you kick something off, it kicks off coenzyme A as CoAsh, and now that acetyl group is now on whatever did the attackee. Okay, so that's one way that you transfer. The enzymes are called acetyltransferases, and they're involved in lots of things. Protein modification, they're involved for lipidation, they're involved in lots of different things. Signaling pathways. Okay. All right, but yeah, you can see here there are lots of functional groups that's involved. This one you can look at on your own. I'll tell you right now, I'm not gonna test you over it. So you take life origins. Life origins. Except for the term monomers and polymers. That's something that you just want to review. A monomer is the simplest of the building blocks. Polymer just means that there's more than one. Okay. So if we think back, proteins would be the polymer, but proteins are made up of what kind of monomers? Amino acids. Okay. What about polysaccharides, which are sugars? Polysaccharides, what is its monomer? Monosaccharide. It's not meant to be a trick question. Sometimes people do. Let's see. I was trying to think of some of the other examples. What about DNA? DNA is the polymer. What is its monomer? The nucleotide. The nucleic acid nucleotide. So that's why it's just some verbiage that's used. Okay, this, just make sure you know, I'm not going to go into a lot of detail here. You should know the parts to a cell. Okay, we're not going to review that at this point. On your own, go through and think about the parts to a cell, what happens there. There's certain parts, I don't want to say are more important, but certain parts that you see over and over and over and over again. First of all, you can ignore cell wall. I skip Untitled Chapter. It's not that it's not interesting, but we just don't have enough time, right? So you can don't forget it. But we don't talk about cell walls. But what do you suppose are some of the really important parts of the cell for in vivo that we're going to talk about a lot throughout, if not this semester, then maybe next semester, but we do talk about a lot. What's one? The nucleus. Okay, the nucleus is important. What happens in the nucleus? What's in the nucleus? DNA. Is that the only place where DNA is at in the cell? No, what else has DNA? Mitochondria. Okay. And chloroplasts. But okay, mitochondria that has it. All right. And then what's another part? Someone said cytosol is actually an important part. We're talking about cytoplasm a lot, especially next semester. It's all about location. What's another important part of the cell you think that we're going to hear about in biochemistry? Pardon?
Cell membrane.
The membrane. In fact, this whole chapter later on, we're talking about membrane trafficking and things like that. What's another part of the cell that's important? There's only two parts of the cell. The ER, okay, this is the rough and the smooth. And the ER stands for endoplasmic reticulum. And what's one of the important things for biochemistry that happens there? And we'll talk about, we actually don't think we cover that next semester. The protein synthesis is the one. Okay, there's also some protein modifications and also some fat modifications, the modifications that occur from ER, which we do talk about next semester. What about anything else? The Golgi, which is, I don't want to say overrated because it's very important, but biochemistry don't cover it a whole lot. Not in this class or next semester, like they are. It has to do with like post-translational modifications and packaging and things like that. So we don't cover that a lot. But you're missing one B, like we cover a lot next semester. There's the ribosome, mitochondria. It's still my hard work on mitochondria. But yeah, mitochondria are hugely important. We have a whole chapter and we talk about that. So make sure you know the parts of the cell. All right, I do usually talk a little bit about this because first for a couple of reasons. One, I worked on it. So you know, if I worked on it, it's going to be important. No, I just wanted to say that. Keep you awake. Keep you on your toes. Keep you on your toes. But also, because I don't know about now, but in life organs, do you guys talk about the RNA World hypothesis? What? Or did you see through it? The RNA World hypothesis. Okay. So, what that has to do, it's sort of like, which came first, the chicken or an egg? There, you know, even though I started 20 some odd years ago, there are scientists that really try to look and say, what's the first biomolecule out there? You know, yes, we've got carbon dioxide, we've got water, and all that. Those are important. But what ones are like, which came first? It has to do with that whole central dogma, which actually I don't have on here, but that central dogma of biochemistry or molecular biology, as they a lot of times talk about, which is what? We have DNA. Oh, whoops, I'm not writing. DNA goes on to make RNA, which can go on to make protein, which can go on to whatever, you know, it comes back to here, it goes on to make enzymes. The process of DNA making DNA was called, DNA to DNA? Replication. Technically it's called DNA replication. The process of going from DNA to RNA is called transcription. From RNA to DNA is called, RNA to DNA. Reverse transcription. Okay. RNA to protein. Translation. Okay. And then what about RNA to RNA? RNA replication. Wow, you get the metaphorical cookie award. Yes. So, but there's all this. And so there's this whole thought process out there. Like, what comes first? Because nowadays, in most organisms, most of the functionality is due to enzymes and proteins, right? Enzymes and proteins. That's not to say that DNA and RNA doesn't have any functionality. We talk about that. In fact, that's chapter 8 or 9, I think. But they look at this, and there were scientists that said, okay, what is the purpose of DNA? What does DNA do? What's its main purpose? Stores information. If you just had to talk about RNA, what's its purpose?
Transport that information.
It can. What else is it? Oh, wow. What's one type of RNA? tRNA. What does tRNA do?
Allows the picking of individual parts that make up a protein.
Okay. Yes, it has to do with the proficiency and also the efficiency, like how efficient it is. Otherwise, amino acids would just be all over. It reads. What does it read? Anthropomorphizing. The what? codons, and codons are on mRNA. Okay, it reads mRNA, brings amino acids and all that good stuff. But RNA has lots of functions. So the RNA world hypothesis is the idea that, it's a hypothesis obviously, that the first true biomolecule was RNA. RNA can actually be, there are enzymes that are RNA does work just like a protein. RNA can replicate itself. RNA can be used to make DNA. RNA can be used to make protein. They can, at least in vitro, and there's, I can't remember his name, Leonidas, I forgot, because I worked on RNA, that his whole purpose, I don't say of living, but his whole purpose of his lab was to prove and to do what's called CELOX, this method of where you deliberately make RNAs that can do things, to try to prove this idea that RNA is the first biomolecule in the bigger scheme of things, and for evolution studies. Versus there's some that said it would have to be DNA, because DNA is the most proficient, or some that said it's really protein and you go backwards from that protein. But no, the RNA World hypothesis is that RNA was the first biomolecule. It was like the whole chicken and the egg kind of argument. Okay, one of the pieces, there are a couple of evidence or facts that they use to try to talk about RNA World hypothesis, and one of them I worked on. Okay, and that was RNase P, it's an enzyme, that every organism, from the simplest, mitochondria even have it, mitochondria and chloroplasts, which aren't even organisms, but the simplest bacteria to the most complex mammals and humans and stuff have to have this enzyme. Okay, however, what the components of it, and it's a quaternary structure for those of you in neurochemistry, the components are very different. On the evolutionary timescale, bacteria, it's the RNA, it has an RNA component, and it has one RNA, it has one protein, and the RNA does the chemistry. The protein just helps with the sufficiency. Whereas if you look at humans and yeast, the eukaryotes, it has one RNA component, and it has multiple proteins, and the RNA alone cannot do the chemistry. The RNA still does the chemistry, but it has to have the proteins there. The bacteria doesn't have to. And then it goes to mitochondria, which what do evolutionists believe that mitochondria came from?
Bacteria?
Bacteria. It's called endosymbiosis, where the cell engulfed. And so it's on the evolutionary time scale that came later, supposedly. And mitochondria actually has a protein enzyme. It doesn't have RNA. So they said, see, look, this shows you RNA being the enzyme, to RNA being the enzyme, it has to have protein, to RNA not even being necessary at all. And so that was one. The other piece of evidence that they like to use is the ribosome itself. Okay, the ribosome. So I actually answered a lot of these on here. But yeah, it's called the RNA World. And that later on, DNA became important. Okay, well, DNA is important. And we'll talk about chapters 8 and 9, the reason why. Like, why do we need DNA if RNA can do all of this? Why do we need proteins if RNA, or why does RNA need? So we talk about a little bit about that as well. Even though maybe it's a little bit more philosophical, but. Eukaryotes, prokaryotes. What's a prokaryote? What's the difference? What is the difference? The presence of a nucleus. Which one has a nucleus? Eukaryotes. Okay. And I should point out the fact that it's a membrane-bound nucleus. Okay, so for the eukaryotes. Then I've got a couple of slides here that just go through parts of the cell. I got them from a couple of different books, like organelles and stuff like that. And so, it's just there to help you, to refresh your memory. Some of these, I don't want to say maybe outdated, but they may have added to since, whoops, that's the wrong one. I was looking in particular, they don't even have it on here. Like, for example, the nucleolus. Where is the nucleolus? It's not even on this list, but where's the nucleolus? It's in the nucleus. Okay, what's the difference?
It's got histones and
Crap. Yeah, that's essentially does it have a membrane? Does the nucleolus have a membrane? Does the nucleus have a membrane? Yes. Does the nucleolus have a membrane? No. Okay, and so there's actually some people that actually don't even believe that it really exists. That it's just kind of like a densely populated area of proteins and nucleic acids that come together. Okay, but yeah, we talk a little bit about that later on. But it's not even listed on here. But on your own, you can just review this. It should be a review for all of this. Okay, another term that gets used are the domains. There's archaea, the archaebacteria, eubacteria, and eukarya. Okay, do you guys cover this? Is it in micro or is it in genetics or biology? Maybe biology. So archae just means literally means ancient. Those are supposed to be the ones that evolutionarily are the oldest. Eubacteria are those that you probably are thinking about when you think about bacteria, like E. coli and things like that. Those are going to be eubacteria. And then eukarya, which are eukaryotes. Okay, just so that way. On your own, you can look at energy, plants. Like I said, we're not going to really cover that. Right. This is not really important. I do want to cover a little bit with ATP. Because we talk about it a lot next semester in particular. I mean, we talk about it a lot. What is ATP used for? Energy. Okay, it is what I call it is sort of like our currency. Okay, just like the stock market talks about dollars and the transfer of funds and stuff. A lot of times we do that with ATP. Okay, ATP itself, with few exceptions, is not the end goal. It's called coupling. It's usually coupled to something else. Okay, so most of the time, the reactions that utilize ATP have at least two steps. And the first step is a phosphorylation or a pyrophosphorylation event. That's a new term that you probably, does anyone know what pyrophosphorylation, pyrophosphate means? Okay, so pyrophosphorylation means two phosphates. This is called inorganic pyrophosphate. It's got two phosphates. It's inorganic, so there's no carbon. It's very important. When it gets hydrolyzed, ATP can be hydrolyzed. There are lots of places that can be attacked, right? You can attack this terminal phosphate, which is the gamma one, and knock off ADP. And so then that's a monophosphorylation event. But many times, because phosphorylation can give off energy, but many times you can also do what's called a pyrophosphorylation, which is where you attack at this point. Technically, you attack either here or here, but you attack at this point and you kick off AMP, which is adenosine monophosphate. And one of the reasons why is that your body likes to do this. The very first true biochemistry professor I had hated it when we referred to the body or your body. He would take off points. I won't if you say that. He wanted you to be specific. Right? So, but your body, whenever it likes pyrophosphate, because we actually have an enzyme that breaks it down further, and you get even more energy. Okay, it's called inorganic pyrophosphatase. And so that's why ATP, and in theory at least, you could also attack it here and just kick off the adenosine, but that in vivo that doesn't really happen. No, in vitro you may be able to do that. In vivo, our body either wants to do single phosphorylation or pyrophosphorylation. Okay. All right, how are we doing on time? So for right now, I'm going to go ahead and I'm going to